Hello, my excellent biologists. This is Organic Chemistry Part 2. This is going to be easier because you already did the hard work where you learned about dehydration synthesis, how we built polymers out of monomers, and hydrolysis, how you break them apart. You've already learned that there are bricks that we can think of as the monomers that build those polymers, and we discussed carbohydrates specifically, and we discussed lipids. So there's two important organic molecules that you need to be really familiar with, and those are proteins and nucleic acids. So that's what we're going to discuss today, and then you'll have this part down. All right, so I'm going to make myself a little bit smaller and go ahead and present to you. So this is Organic Chemistry Part 2. Let me get a pointer in case I need it. So the first thing here we're going to talk about is proteins. So when I say proteins to you, I think a lot of you just think about proteins that you consume, right? You use that as part of your nutrients that you need, right? So, but there's a reason why you need them, and you need them because they play critical functions in your body. So let's look at some of those critical functions that proteins play in your body. All right, so just scan around here a little bit. Things like hair and nails, yes. Muscles you can think of. Hemoglobin's really important. You have 250 million molecules of hemoglobin in every single red blood cell, and every one of those 250 million molecules of hemoglobin, they can carry 4 O2s, so 250 million times 4. 1 billion molecules of oxygen are carried in a single red blood cell. So that hemoglobin seems pretty important. We look at the way our brain and our nerve functioning and cell signaling goes on in our brains and nerves. Enzymes, super critical. We're going to spend some time talking about enzymes later. Those we've already done it a little bit in part 1 and how they catalyze those reactions for us. Cellular construction workers, antibodies to protect you and your immune system, and then, of course, cell messengers. So proteins, super important. So let's talk about the brick, how they're built. The structure of all proteins are out of amino acids. And so you can see, and you're going to need to know this. You're just going to need to know it. You draw a carbon in the center, and then you have 4 lines. My husband is making me laugh. You have 4 lines coming off of the carbon. Now let's follow. When I say lines, I mean bonds, of course. So the simple part is go straight up and you got a hydrogen. Okay, so that's part 1. When you look over here to the right, remember when we talked about functional groups in part 1, this is a carboxyl group. Now this you will see drawn in other diagrams. You'll see C double bond O and then C with a single bond to an oxygen and an H here typically. But when you put it in an aqueous solution, this hydrogen, this is carboxylic acid, right? So it's going to donate hydrogen ions. So it typically loses its hydrogen ion, and where did it go? Dun, 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 right here. Okay, because you'll see this in charts is just your amino group will just be N2, but you can see where the hydrogen went right over here. So when you look at this single amino acid, you've got a negative charge on one end, and then you've got a positive charge on another end. That's going to become important later. Okay, if you looked on your periodic table, you will find no element that is R. R group is like X in an equation. There are 20 different options for R. And if there's 20 different options, that means they could have different properties, right? So we've talked a little bit about that. So let's look at an example or two. Okay, so here are a few examples. Here's alanine. Notice the central carbon, go up, hydrogen, carboxyl group. You've got your amino group over here. And then it's R group, very simple. Just a single carbon with hydrogens around it, Ch.03. Okay, valine, same thing. Okay, but you're bound to 2 more carbons when you look at valine. And let's look at cysteine. Cysteine has a single carbon and then a sulfur group. Now, this will be interesting because there's things called cysteine bridges. So if you have 2 cysteines next to each other, instead of hanging on to hydrogen, the 2 sulfurs may bind with each other. This is phenylalanine. This is not drawn to scale, right? Because each one of these points represents a carbon, and then you can see there, those would be double bonds. So here's 4. This is going to get smaller, so you're not going to be able to see it as well. Here are all 20. Now, what you probably can't see because it's tiny little print, okay? But these 1, 2, 3, 4, 5, 6, 7, 8, 9, these 9 right here, I don't know if you can see, they're just a bunch of different arrangements of carbon for the most part. Or this one, glycine, just has a hydrogen. This one does have some double bonds and an amino group in it. This is tryptophan. You've probably heard about that with turkey, right? And then methionine right here, it's got a sulfur right here. But all of these, okay, are amino acids with non-polar hydrophobic R groups. Hydrophobic, which means, are they going to dissolve well in water or no? Okay, hopefully you said no, because I told you they were hydrophobic. Okay, now your second group down here, let's look at these. We've got carbon and sulfur, carbon and oxygen. We've got carbon and some oxygens down here, an amino group, double bond oxygen, double bond oxygen. Looks like there's an oxygen right here. These are amino acids with polar R groups, which would by default make them what? Yes, hydrophilic, plus it's written right here. Okay, so we've got some hydrophobics and some hydrophilics. Then this third group down here, okay, so you can see COO minus, maybe you can see that. N plus H2, we've got a carboxyl group down here. You can see it's lost its hydrogen, just like an acid would. Here's an amino group here, and here's some other amino groups. So these are amino acids with ionized R groups. So what I want to know is, are ionized groups hydrophilic or hydrophobic? What do you think? Philic or phobic? Hopefully you said hydrophilic, okay? Because they have those charged ends, right? Then water can associate itself. So if it's a negative charge on that R group, then what's going to point towards that negative charge on that R group? Will the hydrogens of the water point towards it, or the oxygens? What do you think? Remember, oxygen is partially negative and hydrogens are partially positive because of the polar groups within the water, right? So if you have a negative R group, you'll have the hydrogens oriented towards it as bound water. If you've got a positive group, then you'll have the oxygen pointed towards it and as its orientation. All right, so those are all 20. Let's talk about how we join monomers, these individual amino acids, to a chain, a polypeptide, and how we do that. And guess how we're going to do it? It's the same way you built fats and the same way you built sugars, right? What was the name of that reaction by which you join monomers? Tell me, tell me, tell me. Hopefully you said dehydration synthesis or condensation synthesis. Remember, if you're going to dehydrate something, what are you taking out of it? Tell me. You're taking water out, right? Dehydrate, you remove water. And that's because each of the monomers, if we're going to join and form a dipeptide, okay, I've got to take something away from each of them so that they'll want to bind together. So let's look at an example of that. All right, so here, let me make myself smaller. I'm in the way. I'm in the way. All right, so here, yes, hide menu. Sorry, it took me a quick minute. All right, so here's amino acid 1. Here's amino acid 2. Now, this is its R group. It's just a single H. And if we go back here, you can see, remember this single H? So that must mean we're looking at 2-glycine. Okay, so these are both glycine. So notice right here, we got to take an OH. We're going to take that right here from this carboxyl group, from the C terminus in, carboxyl group, right? And then we're going to take this H right here from the N terminus end, is where the N, right? N, C. And as a result of that, you're going to form what's called a peptide bond. A peptide bond is a special bond just between amino acids. So it's right here between the C terminus and the N terminus. This is an example, if you remember from looking at the expectations for this unit, is to understand directionality. So this is an example of directionality. You know, you have a left and you have a right hand. Amino acids have an N terminus and a C terminus end. All right. So you can think of it like that. So new amino acids are always added. okay, right here. Okay, good. Now, okay, so if you're going to build a long chain, you always build it off the C terminus end. All right, let's go here. So when you look at proteins, and actually I'm going to make myself bigger because I need to talk to you for a minute. All right, when you make a long chain of proteins, they're made out of single amino acids. So when you just name them, remember there are 20 different amino acids, if you were just like naming each one right there, that's referred to as that primary structure. The primary, the basic structure, primary, is just a single chain of amino acids. Now, I'm going to go see if I can find a pipe cleaner. Hang on here. Okay, I didn't have pipe cleaners, but my husband had some wire, so we'll go with that. Okay, so if you looked at a long chain of amino acids all together, okay, we could just look at this straight piece of wire, okay? But remember what we learned about the directionality, right? And then remember how we looked at that N-terminus end and it had a plus charge, and we looked at the carboxyl end and it had a minus charge? What do opposites do? Yes, they attract. And so proteins will enter into what's called a secondary structure. And that secondary structure is because of the way those opposites attract. And so the two ways they'll go into it is either called an alpha helix, and this is just because of the way opposites attract, or another shape will be a beta pleated sheet. Okay, this is referred to the secondary structure. Something key you need to know about secondary structures. It is not dependent on the R groups, not one bit, not one bit. It doesn't matter what your R group is. The secondary structures, these two shapes, alpha helix, beta pleated sheet, are solely dependent on that the amino group has a positive charge, right? And that the carboxyl group has a negative. Okay, so primary structure, right, is just the sequence of amino acids. Secondary structure, alpha helix, beta pleated sheet, and that is totally dependent on the carboxyl and amino group in the interaction. The tertiary structure is sometimes you'll take this out, for example, this alpha helix. Remember what we had? We had some groups that were hydrophilic and some were hydrophobic. So the philics want to get together and the phobics want to get together. So what happens is this causes it to fold itself so the hydrophilics can be together and the hydrophobics can be together. So this is dependent on the R group. Absolutely, right? Because the R groups determine if the amino acid is hydrophilic or hydrophobic. Okay, so we're going to talk again. Primary structure, sequence of amino acids. Secondary structure is either an alpha helix or a beta pleated sheet, and that is dependent just on the carboxyl and the amino group. And then the tertiary structure is the folding of the secondary structure. So I folded an alpha helix here. That folding due to the R groups. Now, the fourth level, the quaternary structure, depends on if you have more than one chain of amino acids. If you have more than one chain of amino acids and they start all hooking together here, okay, then this would be the quaternary structure of a protein. Okay, now this quaternary structure, it has to be in the exact right shape in order for that protein to have functionality. Okay, so now let's look at this picture I've got going here in the background. I need to make myself smaller. Here I am. Okay, so here are your amino acids, right? Here you can see that C terminus end and you can see the N terminus end. All right, so that's the primary structure. So each one of those balls represents a single amino acid. Now, your secondary structure, you have two choices, a beta pleated sheet or an alpha helix. The folding of the secondary structure, okay, that folding is called the tertiary structure, and that has to do with the R group so that the hydrophilics can be together and the hydrophobics can be together. And then if you have a quaternary structure, it's when you have more than one chain together. All right, so let's put some notes down, what we have so far. So now going back in your group shared notes, which apparently I forgot to do earlier. So on your functions, okay, going back, let me move myself here. So going back all the way to the functions, okay, enzymes, muscles, hormones, antibodies, hemoglobin carriers, channels, and they are found in cell membranes. So I want to make sure you know that they are found in cell membranes. So if you're using my group shared notes, right? If you're just watching this on YouTube, the group shared notes will be down in the description of the video. And so column 1, you'll see yellow highlighted places. That's where you need to type in the notes. Column 2, add in pictures that are helpful for you and your team. Okay, so number 1 on proteins, we're in section 1.4, is carriers and channels in cell membranes, etc. Okay, what are they built out of? Amino acids, that's the structure. You have a central alpha carbon, so here's your central alpha carbon, bonded to an amino group. Here's your amino group, okay? And that's the, I've already identified that as the N-terminus. You have a carboxyl group on this end, that's the C-terminus. And then new amino acids are added to the C-terminus end of a polypeptide through what reaction? Well, we saw that right here. That's the same reaction as before, dehydration synthesis. So through dehydration synthesis, forming, and this is called a peptide bond. Okay, what else do they have? They have a hydrogen, right? We looked at that. And they have an R group. The R group, which determines the identity of the amino acid. So let's go back here. Here are all 20, right? So this R group right here was just an H, so that made it glycine. If it had a CH3 right here, that is alanine. So the R group, which determines the identity of the amino acids. How many different R groups are there? 20, okay? The R group determines the chemical properties of the amino acid. Is it acidic, basic, right? Polar, nonpolar? All right, peptides. Peptide bonds are a covalent bond between 2 amino acids, between the amino group and the carboxyl group. And then directionality is defined, okay? Directionality is defined as amino acids have 2 ends that are chemically distinct. It has 2 ends that are chemically distinct from one another. New amino acids are added to the C-terminus end. And then polypeptides are chains of amino acids. So then we talked about structure. So the primary structure is a sequence of amino acids in chains. You might have 2 chains. Yeah. All right. Secondary, well, let me say primary again because I went fast. Sequence of amino acids in chains. Okay. Secondary is due to charged amino and carboxyl groups creating either an alpha helix or beta pleated sheet. That's what we're seeing right here. Okay. And tertiary is due to R group interactions. R group interactions, you could form hydrogen bonds. We learned about those. Covalent bonds, disulfide, ionic bonds. And then quaternary, oh, more on that, sorry. It's the 3-dimensional folding pattern imposed on top of the secondary structure. The 3-dimensional folding pattern imposed upon the secondary structure. And then quaternary is when you have 2 or more polypeptide chains combining. 2 or more polypeptide chains combining. All right, so how does this folding take place? Right? That's the thing, because if it's not folded in the right shape, it's not going to work. So there are proteins called chaperone proteins, and if you have a normal protein that gets damaged, it'll fix it, right, and try to return it. If it can't fix its damaged proteins, then you got to get rid of it. And so you have proteins called chaperone proteins, which help fold those proteins into their correct shape. If they're not folded in the right shape, lose all functionality. Now, let's talk about some damage of misfolded proteins. And those are, we're going to get there in 2 more seconds. Those misfolded proteins, you might know some as prions. But, and I'll get to that, put a pin in that. Look at this protein here, okay? And then look at this protein here. It's been denatured. Heat can denature proteins. You've seen this if you've ever cracked an egg into a hot pan. The clear part of the egg, which we call the egg white, doesn't become white until we apply heat to it, right? So heat can change those proteins. Now, can you unfry an egg? No, you cannot, right? Once it's fried, you can't make it clear again. Okay, that's permanent damage. Now, some things you can, like you've seen me with straight hair. I just blew dry it and it's terrible. But if I wanted to curl it, I could use a curling iron, and temporarily it would take the heat of my, and apply it to my hair, and I would have a curl in it for part of that day. Okay, but then I've curled my hair 100 times, right? It still goes back to being a weird straightness if I don't curl it, right? So that's something that can be undone, but some things can't, right? So when you run a really high fever, really, really high fever, they're worried about proteins changing here in your brain that could end up causing, right, brain damage. That's why you don't want to run really high fevers, all right? Now, permanently misfolded proteins are called prions, and prions are really bad because they can make other proteins misfold too. So, and there's some diseases that are associated with that. So on your notes for folding, chaperone proteins help them to fold into their correct shape when they are built. Denaturation is when proteins are exposed to extremes in heat and pH. They undergo an irreversible change. And then prions cause certain diseases and they are misfolded proteins. All right, 3 important organic molecules down. We got one left. Okay, so you should be working on that chart I showed you earlier, right, in part 1, where you identify the important organic molecule, you identify the monomer, and then you identify polymers with functionality. So we already did the first 2 in part 1. So here in part 2, amino acids, and then notice I have labeled this amino acid, right? So that I could recreate it if I needed to on an essay. And then for polymers, I've given several functions. All right, check, let's move on. Okay, and let's talk about nucleic acids. So a nucleic acid that probably you hear the most about is probably DNA, deoxyribonucleic acids. But RNA is a nucleic acid as well, and ATP, the energy currency of the cell, the cheta, okay, that is ATP, that is also a nucleic acid. So let's start with the brick. The brick of all nucleic acids are called nucleotides, okay? So nucleic acids are built out of nucleotides. So when you look at a nucleotide, it has 3 components every single time. Let me make myself bigger, okay? 3 components every single time. You have a nitrogenous base, and I'll talk to you about the variety of nitrogenous bases. You have a nitrogenous base, okay? You have a 5-carbon sugar, and you have a phosphate group. What is this yellow phosphate group? It looks like this. See the phosphorus? It's got double bonded to one oxygen and then single bonded to a bunch of other oxygens, okay? Now, this is something we need to get right now. Again, about directionality, it's going to play a big part when we look at the structure of DNA as well as the replication of DNA. So what I want you to do is I want you to look at this pentose sugar right here, okay? So here's the nitrogenous base. Remember, what is here? If nothing is identified, right where it says 1', what is there? A carbon. Hopefully you said that, okay? So there's 5 carbons in this sugar. Carbon number 1, okay? All the carbons are numbered. Carbon number 1 is like the thumb. It is the one always connected to the nitrogenous base, okay? And then do you see how it looks like a house? Okay, and up on the roof of the house, there's an oxygen. So we're not going to go that direction. We're not going to go to the next one because we can't. We run into an oxygen. No, okay, so go the other way. So carbon number 2 is right next to carbon 1, carbon number 3, carbon number 4, and the chimney is carbon number 5, which in this instance is connected to the phosphate group. Okay, so the carbons are all numbered. This has to do with directionality. If I had an eyeball and I was looking down here and I was looking at the edge, I would refer to this as the 3' end down here because the 3' end, the bottom corner of the house is showing. If I'm up here, okay, up at the top above the phosphate, this would be the 5' end. Remember, left and right, N terminus, C terminus with our amino acids, you have the same thing here. You have the 3' and the 5'. Now, why don't we talk about 1' and 2' ends? The reason is that's to the interior, and this is to the exterior, like if you're looking at DNA. All right, so you talk about the 3' and the 5' end. So first thing I want to talk to you about on this, I know, yeah, I'm going to show you another sugar. Here we go. All right, so DNA is deoxyribonucleic acid, and then RNA is ribonucleic acid. So again, we could number these carbons, right? So here's the chimney, so that's got to be 5, right? So over here is where I start 1, 2, 3, 4, chimney makes 5. Okay, then I go over here, 1, 2, 3, 4, chimney makes 5. Now notice this is called deoxyribose and this is called ribose. Deoxy sounds like it's taking something away. So deoxy sounds like I'm taking away an oxygen. Can you see the difference between these two sugars? Hopefully you can. Here you have an OH group, and this deoxyribose, you only have an H. Okay, that's how you can differentiate between those two sugars easily. All right, also, let's go right here. Here are your nitrogenous bases. So you have a total of 5, but in DNA, you'll only find 4 of them, and in RNA, you'll only find 4 of them. So on this part, okay, let me make myself a little bigger. Maybe no? Yes. Okay. First thing we've learned is we have learned that sugars have directionality, right? And the second thing we've learned is that there's 2 kinds of sugars that you find in these nucleic acids. You find ribose and deoxyribose. Now there are 5 bases. Now, 4 are found in DNA and 4 are found in RNA. Now, of these 5 bases, okay, 2 of them have something in common. They have 2 rings in the nitrogenous base, and those are called purines when you have the 2 rings, and then 3 of them only have 1 ring, and those are called pyrimidines. So let's take a look at this picture, okay, and see if we can see that. So here you can see the 2 rings in adenine, and you can see 2 rings in guanine. And then over here at the pyrimidines, see how they only have 1 ring of carbons? Okay, so I always say, poor primitive pyrimidines, they only have 1 ring. Poor primitive pyrimidines, they only have 1 ring. So they have 1 ring in their nitrogenous base. Purines have 2. The purines are A and G. And A, if you had the element Ag, that spells that would be silver. So pure silver, if that helps you. Okay, so A and G are the purines, and the pyrimidines are cytosine, thymine, and uracil. Now, when you look at thymine and uracil, they're right next to each other. DNA always carries thymine, and RNA always carries uracil. All right, that's just something you're going to need to memorize, and I'll show you that in some pictures as well. So let's get some things down in our notes and make sure we're okay. So in general, there are polymers of nucleotides, and we looked a nucleotide, remember, let's go back here, so we're good. A nucleotide is always a nitrogenous base, okay, a sugar, a 5-carbon sugar, and a phosphate. So they're polymers of nucleotides. Now riddle me this. Look at this base right here. Would that base right there be a purine or a pyrimidine, and why? It is a pyrimidine. Exactly. It's a pyrimidine because it only has 1 ring. And then they put a C right here, so it must be cytosine. Must be cytosine because of the 3 pyrimidines, cytosine is one of those. All right. So the function of DNA is it stores the genetic code. Those are your genes, right, that make up your chromosomes. So it stores the genetic code. Now, I haven't shown you RNA yet. It's coming up here in the notes. But RNA, so I'm going to give you the words for it. You may not understand all of it, but I'll get you there, right? That's in a different chapter. RNA allows for the translation of the genetic code of DNA into the amino acid sequence of proteins. Okay? Now, let's think what that is. Your genes are in your DNA. Okay, that's the code. How it is enacted, how it gets done, what it codes for are proteins. And proteins are made of amino acids. So you got to get the DNA code into the action item of a protein. So that RNA helps you do that. So there's mRNA, which stands for messenger RNA, tRNA, which stands for transfer RNA, and rRNA, which stands for ribosomal RNA. And I promise you, I will teach you all the functionalities of those 3 RNAs. Okay, you also have directionality. because you have a 3' and you have a 5' end, like I showed you here. New nucleotides are added to the 3' end. New nucleotides are added to the 3' end of the chain. Nucleic acids are always anti-parallel when they're double-stranded, okay? So one chain will run from 3 to 5, and the other one runs from 5 to 3. Think of it like a two-way road, okay? Two-way road. And I know that's a lot to take in. I'm going to show you the structure of the DNA so you can see that. Okay, so how they differ, we're getting there. We are so getting there. So one way they differ is DNA has the sugar deoxyribose, where RNA has the sugar ribose. So go to your chart there, and you can see sugar, deoxyribose, and RNA has ribose. Okay, the bases that they have, okay, is DNA. You can see I listed them there. Adenine, right here is a purine, and thymine, and guanine, and cytosine, right? RNA has all of the same ones, but instead of thymine, it has uracil. Okay, so you want to have, instead of thymine, there is uracil. All right, let's go look at some more pictures. Okay, so this is a little chart, okay, that shows you that. And I'm going to show you about the double-strandedness in DNA, and there's a single-stranded in RNA. So on your DNA, you can see two chains held together by hydrogen bonds. I'll show you a picture of that, whereas RNA is single-stranded. Now, that one single strand can fold back on itself and get different shapes, and we'll look at that as well. Okay, take a deep breath. You're almost done, almost done. All right, so looking at the DNA right here, okay, you can see a couple different models of DNA. So here you can see a ladder. Now, running up the center of the ladder is the hydrogen bonds. Here is one chain I'm outlining of nucleotides and a second chain of nucleotides, and they're held together by hydrogen bonds. Those hydrogen bonds are between the nitrogenous bases. And we looked at this a little bit when we talked about water, but notice adenine is always paired with thymine, okay? So here's thymine, thymine. Always, always, always. Adenine with thymine, okay, in DNA, and you have two hydrogen bonds. And guanine and cytosine are always paired together. You could remember that because AT spells at, and cytosine has a curve to it, like a C, and guanine, right, with a G has a curve to it. So the two curved ones are always together, and the other one spells at. Now, if you take that ladder, and if you were holding the end of the ladder, and at this end I started twisting it, that's the double helix shape, so a twisted ladder. Think of it like a spiral staircase. And then here's just another model where they're showing you the shape. So when you look at complementary base pairing, okay, where two strands, nucleic acids are held together by what? Hydrogen bonds. A purine, AG, notice there's always one purine with one pyrimidine. And this is why you can keep the diameter of DNA universal, right? It doesn't change because there's always a 2-ring base connected to a 1-ring base. All right, so nucleic acids are held together by hydrogen bonds, a purine, A and G, always with a pyrimidine, T and C. Adenine has 2 H, 2 hydrogen bonds with thymine, and guanine, 3 H to cytosine. Now, I have told you a lot in here about DNA. We are going to revisit this when we go into more detail about the structure of DNA. So that's coming up. Okay, let's look at RNA. RNA is a single strand. Here is a nucleotide, right? We've got a nitrogenous base. We've got our sugar. This time it'll be ribose, and our phosphate group. So here we can see just single strands. Notice there's no thymine, no T. Instead, there is a U for uracil, right? Now, take a look here. Okay, I need to get this thing to go away down there. So notice you have, see if you can follow this here. You can, I know you can. All right, here is DNA. Why? Because I can see two strands held together. And so this big box here on the left, that would be like one nucleotide chain. And over here, this second big rectangle would be the second. Notice they're held together by hydrogen bonds. You've got cytosine with guanine because they always pair up. A pyrimidine, a single ring with a double ring. Notice they are both connected. Look, coming off of here, you've got a sugar. And then up here off the chimney, you've got a phosphate group. Okay, then coming down off this sugar, another phosphate group. So if you have a sugar, basically the outside is alternating phosphate and sugar groups. You can see the same thing over here. Okay. And then in between them, right, coming off carbon number 1 right here is where you have your nitrogenous base. And here you have a pyrimidine thymine with 2 hydrogen bonds with this purine adenine. Now let's look at directionality. Okay, so I'm going to start right here. Here is the nitrogenous base. This is carbon 1, carbon 2, carbon 3. So that's why they call this the 3' end. If you go up here to the top, okay, this is the highest sugar. Here is the nitrogenous base. It's connected to carbon 1. I can't go up here because there's oxygen. So I go down 2, 3, 4, 5. This is the 5' end. Now look at the houses across the street. Remember how we said it's like a road. Okay, they go 2 different directions, right? Like a road. Notice how all the houses look upside down. Can you see that? Okay, so notice how the oxygen is down here instead of up top like over here. You're still going to number the same way. Here is adenine, the nitrogenous base. Here's carbon 1. I can't go this way because there's oxygen. Here's carbon 1, 2, 3, 4, 5. That's the chimney. That's the 5' end. When I run to the other end, here's the nitrogenous base, guanine. Here's carbon 1, 2. This is the 3' end up here. So that's showing you that directionality, and that's going to become really important in a little bit. Okay, so that was a lot of information. Let's look at a third type of nucleic acid, which is just a modification of a nucleotide. It's a base, okay? Here's your base right here. You know it's what? A pyrimidine or a purine? It is a purine because it has 2 rings. It happens to be adenine. That's why this is called adenosine triphosphate. Here's your adenine, here's your sugar, it's RNA, and here's your phosphate group, but you have 2 more phosphate groups right there. These bonds between the phosphate groups are high energy bonds, and if you break one of those phosphates off, you get something like 7.3 kilocalories of energy. Okay, this is the cheta. This is the energy currency of your cells, right? When a reaction requires energy, it can use energy from ATP. Now let's take a look right here as we move from left to right, okay? Notice they have snapped off a phosphate bond and they've created some energy over here on the product side. Remember, how do we break bonds? What's the name of that reaction? Do you remember? Tell me. Hydrolysis, okay? So remember, hydrolysis, we use water. So water has to be put in to put a wedge between those last 2 phosphates in order to release that energy. So that's why there's the input of water. Conversely, if we move from right to left and we want to take this ADP and turn it into ATP, we need to add a phosphate group on, and that will require energy. And folks, okay, hear me. This is why you eat food. Besides it tastes good, you break bonds from the food that you eat to use that energy ultimately to add a phosphate group onto ATP so you have the money that you need for your cells. When you spend the money, when you spend the energy, you got to eat more food. All right? Hopefully that makes sense to you. So on your notes where it says ATP, okay, where it says ATP, it's adenosine. Here, I'll be smaller. Okay, it's adenosine, which is this right here, which is a ribose and an adenine, and then triphosphate, 3 phosphate groups. And the function is a high energy molecule because the last 2 phosphate bonds release energy when broken. And this energy can be used for synthetic reactions, anabolic reactions, muscle contraction, and transmission of nerve impulses. So if we look here, okay, I have completed the chart here now. You have the 4 important organic molecules. Here's nucleic acids. Here's a nucleotide. You have got a nitrogenous base. Notice all 5 are listed here. A sugar, either deoxyribose in DNA or ribose in RNA, and you've got a phosphate group. DNA, genetic code, RNA is about protein synthesis, and ATP is about energy. All right, so this brings us to the end of Unit 1, okay, for APBio. And remember, look at my master hyperdoc. where I have all the expectations, right? And all the information that we learned and all the supports that you might need in order to review this material. All right. I think you are super smart. Okay. Work on making a chart yourself that you could fill out, right? And if you look at all the things that are in Unit 1, let me review that with you. You had the structure of water and hydrogen bonding. So you should be really good with that. Structure of water, polar, right? And hydrogen bonding. The elements of life, we've gone over that, right? We went in our basic chemistry. The introduction to biological macromolecules and the properties of those biological macromolecules. Those are your, right, carbohydrates, lipids, right? Lipids are fats or steroids, and all the modification of lipids like phospholipids that are in cell membranes, right? And waxes. And then we just went over protein function and then nucleic acids. Okay, and the last one is nucleic acids if you're looking through the expectations. I also encourage you to go back and think about this unit and think about all the science practices we employed, right? We learned about chi-square, right, when we looked at data. All right, you're super smart. I love you, and I look forward to seeing you real soon.